box there. Okay, so thank you, Felix, and thank you so much to our colleagues and friends halfway across the world here. Of course, it's first thing in the morning for you, and it's nighttime over here, which is why I've just, as a note to that, made a black background on my slides just to indicate that it's nighttime. But I have the great honor to introduce you to the AIR community. Many of you already know what this is, but I wanted to give a little bit of background about it. So AIR stands for Adaptive Immune Receptor Repertoire Community, or AIRR-C. And as Felix said, my name is Nina Prak. I'm the current chair of our executive subcommittee here on the AIR community. So
I'm not seeing your slides. I don't know if anybody else is, but
can you see it?
Am I the only one?
I can see it.
Okay. I'll be quiet.
Are other people okay? All right. If anyone has a problem, please feel free to interrupt. I can't see the chat, so somebody will just have to interrupt me if they have a question. Okay, so what is the AIR community? It's actually technically a committee within the Antibody Society, and it's our mission to advance and promote the generation, analysis, and standardization of high throughput sequencing data that characterize adaptive immune receptor repertoire. And so those are AIR-seq data. It's a grassroots organization. We have a variety of different stakeholders ranging from wet and dry bench scientists, computational biologists, immunologists, software engineers, ethicists, legal experts, just a wide range of people who've all come together around this common interest and shared interest in AIR-seq. An important feature of the AIR community is that it is open to anyone who's interested in immune repertoire profiling. And so we hope that everyone who's on the call today in this meeting will join us and be part of the AIR community. We very much look forward to working with any and all of you. This is just giving you a brief structure of what the AIR community looks like. So again, we're partnered with the Antibody Society. This is an international nonprofit that supports antibody-related research and development. And we have within that group, we are our own committee. And you can see we have the executive subcommittee, and then we have a series of working groups and subcommittees. And the working groups and subcommittees really form the core of the AIR community's efforts. So what are these working groups and subcommittees? Let me just walk you through a few of them, or actually most of them. So the working groups, one of them is the Minimal Standards Working Group, which we're merging with data representation. This is a group that develops standards that can assure that investigators can share data and the data can be compatible from one lab to another. So this allows us to really share not only data, but metadata, descriptions of the experiments, and allow that for larger metadata explorations. The Biological Resources Working Group is another working group within the AIR community that's important for coordinating the development of reference samples. So here, really the underlying goal is to have biological standards, calibrators, et cetera, that can be used to normalize data sets and compare data from different experiments across different methods. This group has recently conducted an international survey of different standards that investigators are using. And we have a manuscript that we've submitted for that. The Common Repository Working Group is another working group in the AIR community. This group is very much involved with sort of the bread and butter operational aspects of depositing data, accessing it, and sharing it. And actually Felix is going to give a talk about that, so I'm not going to belabor this one too much here. Last but not least, on this slide, we have the Diagnostics Working Group, and this group is very interested in how AIR-seq data may be applied as a diagnostic tool for the clinics. And I'm sure that many of you on the call today are interested in that question, particularly with respect to COVID-19 and whether there can be fingerprints in the immune repertoire for infection and potentially protection or other features of the disease. Other working groups include the Germline Database group. So this is a group that's devoted to trying to improve and generate complete and accurate sets of reference genes for immunoglobulins and T-cell receptors. This group works collaboratively with several other organizations across the globe, and I'm certainly eager to partner with all of you as well in this effort. It's really critical to have a common and shared nomenclature for the germline genes that make up the immune receptors. Another group is software. So software is, of course, very important for visualization and analysis of immune repertoires. And so this is a group that has been working on comparing different immune repertoire profiling analysis tools and benchmarking them, and also creating a compliance badge for tools that have the correct sort of principles of sharing of software and other features to make the software work and work well with one another. Last but not least, we have a legal and ethics group that is very interested in trying to clarify how the legal barriers and the ethical barriers to sharing AIRR-seq data, clarify IP and licensing for software development and other aspects of AIRR-seq experiments. Now, those are the working groups, and those are really the core of the AIR community. Then there's also the kind of the subcommittees, which are the established committees that address continuous or repetitive duties. And those include the executive committee, which is really the leadership for the AIR community, the inferred allele review committee. So this is, again, something that is a recurrent challenge for us in terms of judging the validity of germline genes and allele names and how those get inferred from AIR-seq data. We also have the communications committee, which is very active, and the meetings committee, last but not least, which is quite busy. As you can see from even just the meetings this year, we've had a 3-day virtual conference on AIR-seq and COVID-19 back in September. We have, of course, this pre-meeting with all of you. And we also have a pre-meeting with the Biological Resources Working Group, as well as, of course, our main meeting coming up next week. I want to highlight a couple quick things about what kinds of things really excite us in the AIR community. And I think the underlying goals that we have are really to facilitate data sharing, to accelerate science. And how does one do this? Well, we need a common language and we need standardization for data sharing. We need a way of getting the data into a data commons. So we call that the AIR data commons. And then we need a way of having investigators be able to access that shared data and analyze it. So the AIR community is extremely busy in developing these tools and in promoting a shared series of repositories. So you can see, for example, within the AIR Data Commons, there are several early adopters that are members of the AIR Data Commons listed here. There's several others that are coming online. And we're also working on a partnership with Adaptive Biotechnologies for T-cell receptor data generation and sharing. So these are all very active in terms of data sharing. And again, Felix will highlight more of that. Also, in terms of data analysis, there are many different analysis tools that are in the works. Again, we have a lot of things going on, and we're very eager to partner with all of you to further promote these aspects of data sharing and analysis. The aspect of data analysis, I just want to highlight that as AIR-seq data become more complex, and we have an explosion really of single cell methods and different multi-omics platforms, the need for really good analysis tools that can not only drill down on single cell data, but also can integrate data across different analysis platforms will become increasingly critical. And so this is something that, with the advent of single cell technologies such as, CITE-seq or DNA bar-coded antigens, LibreSeq, as well as, of course, single cell RNA-seq, epigenetic analyses and other platforms, being able to integrate those and visualize the data will be very exciting for science, and is certainly something that is actively being pursued all around the world. The other thing that goes hand in hand with that are tools that can allow us to look at patterns in immune repertoires, not just within individuals, but across individuals. So visualizing the landscape of the immune system and then looking for fingerprints of disease, whether that be cancer or exposure to COVID-19, are certainly areas of active research and interest to the AIR community. So what can we do together? A lot. I think we can certainly continue the expansion of the AIR Data Commons, and we would love to include you in that partnership. We can continue to work on standardization of nomenclature for data, and we would, again, be very eager to work together with you. Germline allele discovery, inference, validation. There's a lot of work that needs to be done. Promoting the development of data query and visualization tools, developing standards for AIRR-seq experiments, and of course, continued communication, promotion of open data sharing. These are things that we're very, very eager to do. And of course, last but not least, convening international meetings and virtual conferences. And I just, again, want to highlight all the AIRR-seq meetings that are currently happening. So there's, of course, this meeting, this very exciting meeting that's just kicking off here. But in addition to that, we have the Biological Standards and Workflows mini symposium coming up in just 2 days. And we also have the AIR community, the main meeting coming up December 8th through 10th. So the AIR meetings committee has been very active. And also, for those of you who are not yet members of the AIR community, it falls on me as the AIR community chair to warmly welcome everyone and encourage you to join us. So again, please consult our website, and there's information there about how to join, and there's information about benefits of membership to the community. But again, the key point here is that it is an open community, and everyone is welcome to join the working groups, irrespective of whether you're a member. So last but not least, this was a photo from somewhat happier times when we were not all wearing masks, when we were in Genoa together last year. But please visit our website, it's listed there. And there's far more information there about our working groups, subcommittees, and other efforts that are ongoing. So last but not least, I want to thank all of you, and I hope that you will enjoy the rest of your meeting. And this is an owl that tells you that it's almost time for Nina to go to bed. So with that, I will turn it back over to all of you.